But as the trial went on, we came to see that there was a decline in some of the patients' ability to ventilate independently. And ventilator use or any kind of ventilatory assistance was really one of the assessments in the trial, but there were no pulmonary function tests per se done in this patient population. So I venture to say, based on what we know now in the animals, that I think, in fact, all the patients in this young age group, if tested by some provocative measure, would have a deficit in ventilation. It's really a physician-driven decision at what point, and it's difficult to do pulmonary function tests in infants and young children, at what point there is really reached the compensatory mechanisms have failed and you've gone from a compensated ventilation to decompensated, and that's when physicians usually institute some form of ventilatory assistance. That point of respiratory failure is really too late. In many of the patients, we'd like to manage them more in an anticipatory fashion. And I think, going back to the animal model helped us figure that out. So we had really never observed, at least clinically, any abnormalities in the animals that we helped generate with Nina Rabin over many years. And it was really through the help of one faculty member who works on control of breathing at the University of Florida that we started to use some more sophisticated techniques. So we went back to the mouse and we started by looking at the diaphragm. Well, certainly the GAA deficient mouse has glycogen in the diaphragm muscle, and that's not in the normal diaphragm. We also quantified glycogen in the diaphragm, and you can see the glycogen is quite high. But the respiratory system is composed of more than just the diaphragm. There are other muscles that function for respiration, and the muscle of the diaphragm actually needs to fire with a very complex mechanism that controls breathing. And it's really, I originally took it to be as simple as the heart firing and beating every time it does is rather automatic. The diaphragm function and the control of CO2 is a much more complex process, which is even now still poorly understood and significantly influenced by, we heard a lot about how sleep impacts on tone, muscle tone in general, and particularly on breathing. So it's really a lot of steps involved in every breath we take, and it's completely subliminal. It happens automatically. So it's really an amazing system. When we looked specifically at the muscle strength, the diaphragm, as animals age, we found that when you test just the muscle outside of the animal by testing its force, its contractile force in an apparatus, you can see as the animals age, diaphragm function declines. So, and that is true of other skeletal muscles too. We published that a couple of years ago. There's really an age-dependent loss of muscle strength. When we looked at the spinal cord, the other part of the system, particularly in the region of the cervical spinal cord, which participates in control of breathing, we saw that there's also an age-dependent increase in glycogen content. So the control animals have a fairly fixed amount of glycogen. It does increase slightly over age, but in the deficient animals, in the Pompeii animals, particularly in this phrenic area, there's significantly more glycogen. And interestingly, this is not true in the brain. There's quite a differential amount of glycogen in the brain versus a metabolically active tissue like the spinal cord, which has to work continuously and particularly while we're asleep. So this is a fundamental difference of the spinal cord. So we again, in collaboration with Nina, made a mouse model which made GAA in every muscle, but not in any other tissue. So they had really normal GAA activity in all muscles, and particularly the diaphragm. And when we looked at those animals, their strength at a year of age, their strength was very much like the wild-type mice or their controls. So this didn't explain then, when we looked at these animals, why they were also defective in breathing. And the way we assess that is with this apparatus. The mouse equilibrates itself in this little chamber by a method called whole body plethysmography. And this can be used in humans, and it is actually for some other types of respiratory assessments. But we built this little device to be able to control everything about their breathing environment so that they can be conscious and moving around in this area, and we monitor the gases in the chamber. And the mouse's movement actually helps us determine these parts of the respiratory cycle. You can actually measure the changes in lung volume and the flow velocities that occur during either quiet breathing or stimulated faster breathing. And when we looked at the Pompeii animals under this protocol where we allow them to sit for about an hour to equilibrate in the chamber and then expose them to higher CO2, in most people this stimulates faster breathing or higher minute ventilation. And, and that's what we assessed in the animals in multiple age groups. And the take-home from that is that the Pompeii animals had very defective response to hypercapnia, which is shown by the dashed line. So a normal animal starts picking up their respiratory rate, and you see they can double both their tidal volume and their breathing frequency to increase their minute ventilation. And this was not true in the GAA deficient animals, and this in fact was not true in even the ones who had normal GAA in the diaphragm. So this was still puzzling. So when you can correct the ventilation to the CO2 level, because we also found that all the diseased animals had elevated CO2, like many of the patients, you can see usually this ratio represents some abnormality in control of breathing, either from the brain stem or in the other components of the signal, the motor neurons. So another indicator of the deficiency chronically in breathing in the animals is to look at their blood hematocrit and hemoglobin, and you see when you control for other things in the blood chemistry like sodium and glucose, the diseased animals had a much higher hemoglobin and hematocrit, which represents the kind of body's attempt to compensate for this defective ventilation over time. So then we, in a subset of the animals, looked specifically at the phrenic nerve function. And we did that by controlling CO2 and the blood gas in the animal, and then isolating the phrenic nerve and measuring its output. And you can see that in the control animal, every burst of breathing, so the nucleus of the phrenic activity in the spinal cord actually works to try to gauge how much of a breath one needs to take. The big, deep breath fires a lot of motor nuclei, and the little quiet breathing just a few. But each burst looks like this when you look at it up close. A few start to fire, and then over time they cooperate with each other in order to take a full breath. And we found that it was almost absent in the Pompeii animals, meaning that most of the breathing was in fact coming from the extrathoracic musculature and not only the diaphragm. There is some activity of the diaphragm, but really not sufficient, obviously, to manage breathing at all or respond to hypercapnia. And when we looked in the animals, and actually there's been really a very elegant study done similar to this by some of the folks at Genzyme who are tracking very closely the characterizing the animals. You can see that in the anterior part of the spinal cord, right here, that when we label motor neurons with a gold stain which tracks up the phrenic nerve, that they are very dense in PAS staining or for glycogen. In the controls, that's not the case. And in a Pompeii patient who had succumbed to respiratory failure, in an autopsy, we found the same kind of glycogen accumulation in spinal motor neurons of the cervical spinal cord. This also demonstrated a lot of disease within the motor neurons that is suggested by the fact that the nucleus is off to the side and the cell really looks like it's dying. We don't know whether these, and this cell not as bad, we don't know whether these cells actually just have dysfunction or they actually, over life, some of them are eliminated, as happens in other lower motor neuron diseases.